So would you prefer the dimming? Yes. I don't know if I will make sure that Emily and Olivia have access to that so that they can grab it if they need to. Okay. Yeah, yeah. So the lab manual, it's out of print, unfortunately. They don't plan to do a new edition. So we're going to use it for the last time this semester. So if you have a hard time finding it, we will be providing PDFs of that. Okay. All right. So with the cell wall, we looked at the idea that some plant cells have two cell walls, a primary cell wall and a secondary cell wall. All plant cells have a primary cell wall. So that's what they're born with. And so those are more flexible. They're strong. You have those long chains of glucose molecules that are clustered together in microfibrils, and then the microfibrils are clustered into fibrils. So you do have that really strong primary cell wall. But then the secondary cell wall, sometimes some parts of the plant body will make that decision, so to speak, to have a secondary cell wall that gets laid down inside the primary cell wall. It's thicker, it's stronger, and it has lignin. Lignin is unique, it doesn't show up in the primary cell wall, only in the secondary. Really, really strong. And it allows for structural support because plants don't have bones like we do, so they need some other mechanisms in play for the structural secondary cell wall will form. Now, if that part of the plant forms a secondary cell wall, after that cell is mature, the cell's going to die. The cell dies, so it becomes a hollow part of the cell. And that might sound like a bad thing, but it's actually a really good thing for various parts of the plant. Because we're going to learn how the way water moves through the plant is moving that cell that's stacked together, but it creates a very easy way for water to go to the plant body through those hollow cells. Okay, so know the difference between primary and secondary cell wall. And by the way, if you've noticed, if you pull up my PowerPoint, click on the notes view, you'll see that I have typed up extra things in the notes view, right? Have you noticed that? So that's content that's expected for you to know, all right? And so I have it in the PowerPoint. It's just I don't necessarily always put everything on the visual you're looking at in here because I want you to listen to me. I don't want you to try to jump ahead, and I don't want you writing everything down. Remember, they're all available to you. So the stuff that you see, don't focus on writing that down. Focus on writing extra notes that as I'm talking. Okay. So make sure you're looking in the notes view of the PowerPoints because I do have some information there that is necessary. Okay, so that lignin makes it really strong, and it's a very complicated. My goodness, I did a 2-hour meeting this morning, or I didn't do any talking. So lignin, by the way, if you've ever driven by a paper making company, you smell that weird smell in the air. They have to use really strong, smelly chemicals in order to create paper that can get rid of some of the lignin in the process because lignin makes paper pretty brittle. And so breaking down lignin, it's a very strong, complicated polymer. And so they have to attack it with very aggressive chemicals in order to do that. Okay, then we have to think about if you have a tissue, many cells congregated together, you need a way for them to communicate with each other. So neighboring cells have to have a quick way to do that. And that is through the plasmodesmata. That's plural. If you just talk about one, you would say plasmodesma, but I hardly ever say that word. I always talk about it plural. So plasmodesmata are the tunnels that link up to neighboring cells. And they're open. There's no membrane covering the opening. There's membrane material that lines the side and goes from one cell to the next. But it's all open. It's like a tunnel. And that allows for small molecules, small chemicals, small proteins and enzymes to flow hormones sometimes, they flow from cell to cell through the plasmodesmata. So very important mechanism for that communication. So here you're seeing on the far left here, that picture, those dark bands, those are the plasmodesmata. And then they have a colorized drawing of it where they're trying to show some of the detail. Okay, so plasmodesmata, please get that term in your head for the tunnels that connect to neighboring cells. Okay, now let's look at some of the interesting organelles that you would see in many plant cells. Of course, the chloroplast is huge. You learned about that probably since grade school. And we will come back to this in a little bit more detail when we talk about photosynthesis, but let's introduce it. So the chloroplast is interesting because it has two membranes that surround it, so-called outer membrane and then an inner membrane. And then if you were to crack open and look to the internal contents, you'd see yet another membrane. All right, and so that internal membrane, which they have the word there, it's a little small, right there, thylakoids. Thylakoids. So the thylakoid is the internal membrane, the green thing that you're looking at there, all right? So all of that taken together is called the thylakoid. Now they break down the thylakoid into some distinct, they give some more terms because there's some distinct regions. You see how some of the thylakoid membrane is stacked into, they look like little discs that are stacked. That's all membrane material. It's just stacked up like that. And when you talk about that, you refer to it as the grana. Or if you want to use a singular word, it's granum. Granum. I usually don't say singular, I just say grana. You put an A on the end and then that's plural. So the grana or the singular granum, that's those stacked regions of the thylakoid membrane. And then, I don't know if they have the word on here. All right, I'm going to write it down. So then if you look at the relatively unstacked region, so I'm going to circle right there. That relatively unstacked region, that's kind of like the bridge between hooking together two grana. That you would call the stroma lamellae. L-A, oh my goodness. I'm going to go to the last slide where I have more space. Oh, I thought I had one. See if I can do it here. Just ignore this slide for a second. Stroma lamellae. L-A-M-E-L-L-A-E. So the stroma lamellae are the relatively unstacked regions, okay? The relatively unstacked regions of the thylakoid membrane, stroma lamellae. Now, why do they give you two more jargon words to memorize? It's because some stuff happens in the grana region, that's physiologically during photosynthesis. Some stuff happens in the grana. Some stuff happens in the stroma lamellae. So they had to give it two different terms so that when they talk about it, we know what's going on. Okay, so let me go back. All right, so the stroma lamellae are the relatively unstacked regions that you see where I circled it here, the little bridge between the grana. Okay, so I would like you to commit those terms to memory. All right, typically that kind of thing would be a multiple choice question where I would describe it or ask you to define, you know, the grana or the thylakoid, etc. So that's a site of photosynthesis, of course, because all that green, by the way, of course, is where the chlorophyll pigment. So there's lots and lots of chlorophyll pigment embedded in that thylakoid membrane. And the chlorophyll pigment is what is responsible for absorbing that sunlight energy to allow for photosynthesis to occur. The gray pictures here, by the way, on the left, those are actual electron microscope pictures of chloroplasts, and you can see the stacked and relatively unstacked regions of the thylakoid membrane. So chloroplasts are a type of plastid. Plastids is like the umbrella term for organelles, and then there's like subtypes of plastids. So there's chloroplasts is one famous example of a plastid. But there are also things called chromoplasts inside many plant cells. And you'll get to look at them today under the microscope. This is tomato flesh put under the light microscope with images being seen here now. And you can see all the little red blobs. Those are chromoplasts. All right. And so that's what makes the tomato half red. It's the pigments inside those chromo. So chromo means color. Other than chlorophyll, so it's a separate from green, a chlorophyll idea. So chromoplasts, you can have some chromoplasts that are red, some that are more yellowish, purplish. It just depends. Yes. Carotenoids. Yeah, carotenoids are pigments too that are giving, like carrots are the prime example. If you eat a carrot, you're eating a lot of carotenoids. Beta carotene is a carotenoid. And so, but in this case, with the chromoplast, you can see, I think you will look at carrot tissue today in lab too. So you'll get to see that in the lab today. I think we have available tomato, carrots, some different colored peppers, like bell peppers, the green, red, yellow, can't remember what else. But so you're going to see several different types. And so the carotenoids, they can be various shades of red and orange and colors like that. Okay, so you'll also look at leucoplasts. Leucoplasts. Leuco is generally saying that they're colorless. They're not that bright. What we just saw with those chromoplasts, that really deep pigment. Here you're looking at leucoplasts, a special type of leucoplast called amyloplasts. A-M-Y-L-O, amyloplasts. So here you're looking at this idea, A-M-Y-L-O, and then put plast on the end, amyloplasts. That's what you're looking at here with amyloplasts. Their job is to store starch. If you look up the word amylo or amylase, that's an enzyme that can break down starch. So that AMYLO is referring to starch. All right, so white potatoes that you get French fries from, but baked potatoes or whatever, that's what you're looking at here on the left. Those, stain the amyloplast so the membrane takes up the purple color. All right, so all those purple kind of egg-shaped organelles, those are amyloplasts. And then on the left, and then on the right, what you're seeing is avocado. So avocado, they don't really have amyloplasts. They have what are called elioplasts. And again, this is in the notes section. So if you pull up the notes, you'll see this. E-L-I-A-O, and then the plast on the end. Elioplasts. Elio is referring to fats. All right, so avocado flesh, you're going to have some of that today too, that you'll squish onto a slide and look at it. And you'll see that, especially when you do it yourself, you're going to see that the flesh of the avocado is just jam-packed with elioplasts. So that's why, it tastes so good. Guacamole, it's very tasty, but it is high in fat, but it's good fat. It's just you need to eat it in moderation. So be careful about that. So you get to see that under the microscope today. Okay, so they have various types of plastids depending on the plant species and which part of the plant you're thinking about. The other idea to remember is that all plants do have mitochondria similar to us. They do the same thing with their mitochondria that we do. All right, and so the mitochondria is involved in largely it's the production of ATP. Okay, ATP is the chemical energy that living organisms on Earth use. And so the mitochondria in plants are doing very similar things to how our mitochondria run and are producing ATP. So very important. Okay, and then another organelle of the plant that is different compared to us is that we don't really have vacuoles, certainly nothing like what a plant does. Plants have very large vacuoles. So a mature plant cell, a baby plant cell will have a little that has to grow for a while. But a mature cell that's all done growing is going to have a whopping big vacuole, and that's a good thing. So the vacuole is the site of turgor pressure. We talk about turgor pressure, water pressure. So what's happening is if you crack open the vacuole and look, it's going to be filled with mostly water, but it is also a storage site for pigments. Sometimes pigments are in there, enzymes, sometimes toxic things that the plant accidentally gets exposed to. They'll pack it into their vacuole. Sometimes they store it in there to try to sequester it away from more important stuff. So there's a variety of things that can be in the vacuole, but water is the number one ingredient. And if a plant is well watered, its vacuole will be full of water that's pressing against the membrane of the vacuole. The membrane is called the tonoplast, T-O-N-O. The tonoplast is the name of the membrane. And so the water is going to be pressing up against that. And if you have a mature plant cell with a whopping big vacuole, that's going to create pressure against the cell membrane and the cell wall. And so this lower picture is trying to get across, you're looking at one cell that has a really big vacuole, it's well watered, and you can see all the other organelles are kind of squished off to the side. So sometimes when you look at a well-watered plant under the microscope, you may have noticed, like, why is everything squished off to the side? Unless you do a very special staining procedure, which is kind of a pain, so we don't really do it, but you can stain the tonoplast, it's just really tricky. But if you see this big empty space when you're looking at cells today or through the semester, if the most of the cell looks empty, it's not. It's the vacuole taking up all that space. So just keep that in mind. Everything will start to look squished off to the side if it's a well-watered plant. So what's happening is water's rushed into the vacuole, that's pressing against the tonoplast membrane, that's pressing against the cell membrane, that's pressing against the cell wall, and so you get this turgor pressure. And again, that's a good thing. Plants don't have bones, so this is another way that they can try to stay upright and have some structural support is through turgor pressure, water pressure, as well as, you know, the cell walls that they have. So it's a good thing. Okay. Well, let's zoom out a little and think about the plant body in a little bit more detail. So there's some anatomy that I'd like you to start learning. And so here you're looking at the overall plant body. It's kind of a, when you think about plants, how they live their lives, the typical plant sticks that roots in the soil, it's a very different life compared to what's happening above ground versus what's happening below ground. So they're very unique organisms that have to live half their bodies above ground and more than half their bodies under ground. And so it's next time you're out walking in the woods or working in the garden, keep in mind that what you see above ground is actually less of what you see in biomass, overall biomass. There's more root biomass than shoot biomass. So there's more underground. Just don't see, of course, it's in the soil. Same with your walking out in the woods. As you're walking through the forest, the root systems of those plants is far more massive in terms of overall biomass than what you see above ground. All right, so it really is a hidden life underground there. So you see here the shoot, you have the stem, and you can see that they're trying to give you a couple of things to think about with the stem tissue. You have the node region. The node region is where branches or leaves attach to the stem. That's called the node region. And then in between those areas of the stem where you don't see a branch or a leaf attaching, they call that the internode. Inter means between. So node region is where the branches or leaves are. Also noteworthy, when you look at the node region, you will typically find axillary buds. So I need you to know that term, axillary bud. The axillary bud is important because it's a site of active growth. Not continuous, like right now, this axillary bud is dormant. It's not really growing, it's just waiting until it gets the signal. But once it does get this signal, and we'll talk more about that, what that means, that axillary bud will start to grow into a new leaf or a new branch with leaves. Okay, so that axillary bud is a region of the body that's primed and ready to grow when it gets the signal, and then it can kick into very active growth. Yes, you got it. So we'll talk about how that happens in the next slide set, actually. So yeah, hormonal control keeps axillary buds quiet and also kicks them into high gear. So the axillary buds, you're actually going to need to know this information, by the way, in lab next week when you do some propagation. We're going to go to the greenhouse and you're going to learn how to propagate through stem cuttings, maybe somebody's cutting as well, and learn how to clone plants through propagation. And we'll be telling you, look for along the stem area, look for the node region, try to find axillary bud, because then you have a really high chance of success where you're going to get a good propagated plant in the end. So, okay. What else? Then at the very tip, you see the shoot apex. Shoot apex. That's another very important part of the plant because there's a cluster of cells there that, again, are primed and ready to grow. And the shoot apex actually is pretty active through the life of the plant, especially when it's young. So the shoot apex has meristematic tissue that allows for continual growth when given the signal. Very active growth. So those are very precious cells. And you know, if the plant gets that part of their body damaged, it can cause some problems. It's not the end of the world, but it can cause some problems. So those clusters of cells, here's the actual word for it, meristematic tissue. Okay, so the shoot apex, the tip of the shoot, but also the root tips. Okay, so both the shoot tip and the root tips have meristematic tissue. We call it apical meristem tissue. Active clusters of cells that are actively growing. Okay? And so today you're going to get your seed germinate. And so what you're going to do is activate that seed to tell it start germinating, start growing. And on Thursday when you walk into lab, hopefully you'll see the embryonic root, which is called the radicle. That will be coming out of the seed. And it's because of that root tip growing, and came out of the seed and is searching for that precious water. So the question is, what happens if you cut off the top of a plant? And you can do this. So if you cut off, and I'll talk more about this in the next slide set too, and on that topic, if you cut off the shoot tip, there's a hormone there in the shoot tip called auxin, that if you cut that part off, the auxin goes away in that area. And it's actually the auxin that is keeping all the axillary buds below it quiet and dormant. But if you get rid of that auxin, now those axillary buds don't get the signal to be quiet anymore. So cutting off the shoot apex will actually make the axillary buds grow. So you get growth. It's just you get a more bushier growth rather than just straight up growth. You'll get the axillary buds activating and you'll get a bushier plant. Yeah. To get more of, yes, and more options for flower buds. Yeah, absolutely. It's a very good strategy. Effective, it's simple. You know, just cut off the top. I do that with my house plants. I don't have a ton of room. I don't have a very big house, and I don't have a lot of room by the windows that are adequate for plant growth. And so I often get leggy growth because they're looking for more and more light. So I often will do that strategy to try to keep them from growing too tall and get them to bush out a little bit. So, okay, what else? So then, oh, here with the roots, you see tap roots. So this artist drew this kind of root system where there's a defined bigger tap root, and then you'll have lateral roots. But there's different variations of root design. This tap root is a good strategy, but there's also the fibrous root system. You'll learn about that in lab as well. You'll get to see some examples where if we say it's a fibrous root, that means there's no central tap root. All the roots pretty much the same diameter, same thickness, same length in general. Yeah, root systems are amazing. Like sometimes when I propagate plants, well, dividing plants, I guess is the better way to say it. When I divide plants, like I'll cut through, especially like my African violets at home, they all grow their pot. And so I'll just uproot the whole thing and just kind of cut it in half, including the roots. And the plant can survive that and do quite well. So you can cut, and you know, if you cut all the root apical meristems off, that's probably not the best thing to do, right? But if you cut it more like down that way, like right through the middle, that would be okay. So it just depends on the root system. Yeah, yeah, absolutely. Yeah, in fact, hydroponics, growing plants hydroponically, and then we had a beautiful, huge, I don't know if it 20-gallon, 40-gallon fish tank, and we created an herbarium in it with small little hoses for water. It was the back wall we designed. It wasn't me, it was my grad student. He had too much time on his hands apparently, but he made this beautiful herbarium with the back wall was a waterfall. So he had hoses, hidden water hoses that were trickling water. But what happened was all the moss that he had put on there, their roots went right into those little tiny hoses, clogged all the water hoses. We came back, you know, so that happened during break. We came back in the winter semester and the whole thing was dry, everything dead. It was so sad. So as we speak this semester, I have a group of students that are working on trying to get that back up and running. And we're going to put it in the atrium over in Weston for everyone to enjoy. It was when we had it there, people would sit there and read and just stare at it. So I'm hoping we can get back to having that available. So that apical or apex region, those are the tips of the shoot and active area of cell growth when given the signal. All right, some other, so axillary buds are also meristematic region. Okay, the axillary buds are meristematic regions. And then we also have to think about plants who have significant growth in the lateral direction. So big, huge tree trunks that you think about. Those we often refer to as secondary tissues. And you could say it increases the diameter. Often we refer to them as lateral meristems because it's giving rise to that increase in diameter rather than length of the body part. And so think about those massive giant sequoias, massive redwood trees, and those stunning large tree trunks. And so the vascular cambium is the meristematic tissue that gives rise to that increase in diameter. Those beautiful giant trees. All right, they do it for those normal size trees too, but if you think about stem tissue and the increase in diameter, that's the vascular cambium. And the vascular cambium if you blow this up. So this is an image of the bottom right is a cross section of a stem of a younger tree. And then you can see what they're trying to show is as it grows, what's happening inside that tree trunk. And the vascular cambium is the location where you see this shift from the lighter purple area into the dark pink area. So let me get my laser pointer. If I use my laser pointer, it automatically shrinks all the images. It's weird. But that area, that band that you see that shows the shift from that kind of light purple to the dark pink, that ring of cells right there, that's called the vascular cambium. And we'll talk more about it when we get into stem tissue. All right, so this is going to come back, but I'm just introducing it right now. And then when we get into bark, when we start talking about bark tissue, what makes up bark, you'll also think about that. And it's kind of hard to see, but there's a band of cells, another ring of cells on this image. Can you see, if you work your way from the outside, you see the really dark pink, very outermost ring, but then do you see that next pink ring of cells? So this dark band of cells, can you see my pointer? So that ring of cells is the cork cambium. Okay, the cork cambium. So that's another ring of cells that can be very actively growing. It's meristematic tissue. So as you can see, there's different parts of the plant body that is devoted to, I can really actively grow if I'm given the signal, all right? And then other parts of the body where you don't have that tissue. So it's discrete regions through the plant body. So we'll talk more about all of this again as we work through root, stem, seed anatomy, okay? And then grasses are interesting because they have what's called intercalary meristems along the blade, in the blade of the grass. And so that's just another area again of active growth. So that's why you always have to mow your lawn on a regular basis, because those intercalary meristems, you mow your lawn and the intercalary meristems go, okay, I'll grow again. And they produce more of the grass tissue, okay? So grasses have that special intercalary meristem. Okay, so getting at some of the tissue regions that are really special. Now let's think about parenchyma tissue. So parenchyma cells make up parenchyma tissue. It's the #1 type of cell in the plant body. It wins. So if you were to rank it out, you know, what type of cell is the most abundant in the plant body? It would be this, parenchyma. Parenchyma wins, okay? So you're staring at an image of parenchyma cells under the microscope. Nothing too remarkable to look at. They're living cells, of course, full of the typical organelles for that part of the plant body. They have a primary cell wall only. If they're alive, by definition, that means they only have a primary cell wall. They don't have a secondary cell wall. Okay, so they're living cells with a primary cell wall. And they often say they're 14-sided if you count the kind of the all the different sides. Not that you need to know that, but it's just you know, they tend to be fairly large, thin cell walls, super abundant. CW is cell wall. Okay, so parenchyma cells make up parenchyma tissue. Parenchyma tissue is very abundant through the whole plant body. Here, you're looking at collenchyma cells that make up collenchyma tissue. These are more discretely found in various parts of the plant body. So probably the best example I can give you is if you've ever eaten celery stalks and you bite into the celery, and very often when I bite into a celery, you get those strings that it's like, darn it, I didn't bite all the way through. Those strings, the reason that they're hard to bite through, it's parenchyma. Those ridges that you feel along the celery stalk, those are a clustering of parenchyma cells. And so this lower left image that's showing that red outline, they're just trying to outline the region of collenchyma cells. So that's like the ridge of the celery stalk. And they cut a celery stalk in cross section, and you're looking at one of the ridges. And then if you zoom in some more, you can see that they kind of look, I just say odd, in the sense that they seem spaced far apart from each other. There's a lot of this, like the brown space in between the cells. They're living, but they do have thickened up cell walls. So they look like they're kind of spaced out from each other. All right, and they tend to be clustered, like that example in the lower left that makes up the celery ridge on the stalk. So yes, they're still alive, so they don't have any secondary cell wall, but their primary cell wall is thickened up a little bit. Good structural support for the celery stalk. It's a good way to make sure the celery stalk stays upright through the growth of the plant. Okay, so it's just another way to add structural support. You know, so we talked about primary cell wall, secondary cell wall, vacuole with the Turgor pressure. Now we're talking about parenchyma, clusters of parenchyma that can also add to structural support. Okay, so now sclerenchyma tissue. Of course, they have to come up with these big long words that are hard to pronounce. Sclerenchyma tissue. All right, so now we're talking about sclerenchyma cells. Sclerenchyma are thick, tough, and they have secondary cell walls. They are alive or dead. They're dead, but they're still very important. So sclerenchyma cells are dead. Collenchyma that we just learned about, they're alive. All right, sclerenchyma are dead. So, but they're important. They're important in supporting various parts of the plant body, giving some extra structural support. Now, what you're looking at in these images is a special subtype. So there's a couple of different types of sclerenchyma cells. One is called scleride. You're looking at these. You can see they look really different from each other. I mean, the one on the right kind of looks like a starfish. Those are all sclerides, kind of like that starfish look. Here, though, these are with the big, thick cell walls that have those stripes in them. So you can see how big the secondary cell wall is. All that stripy area, that's the cell wall. And they're dead. They're just hollow, okay? But they perform some nice support. The best example I can give you that you've experienced, hopefully, sclerides is if you're eating a pear fruit and you bite into the pear and you feel that really feel against your teeth. Those are sclerides. You're eating a ton of sclerides with each bite, okay? But that's okay. It's a good thing. And so you tend to see sclerides, if you look at them, where are they? They tend to be scattered, like all in the pear flesh, for example, or sometimes we'll see them making up the seed coat of a seed. You'll see a fair bit of sclerides in there. They're hard to define. They look very different depending on the plant species. So they generally just say, oh, they're about as long as they are wide. And that's about as specific a definition. Yeah, because they do have different morphology when you look at them under the microscope, depending on the species. So sclerides are kind of scattered. Here's the other type of sclerenchyma tissue or cell type, fibers. So sclerenchyma is composed of sclerides and fibers. Okay, sclerenchyma, then sclerides and fibers. Fibers are, by definition, they're much longer than they are wide. Any fiber in any plant species is going to have that form where they're long and skinny. All right. So what you're seeing here on the left image is they took a plant part, and cut it in cross section. And now you're just seeing the ends of the fibers, like you're looking down through the fiber. And so you can see a whole cluster of these, all these pink cells are fibers. And you can see the little white dot in the middle, that's the hollow part of the cell, the part that died off. So they have thick secondary cell walls again, dead at maturity. The right image is where they took a cluster of fiber cells and so if my fingers are fiber cells, what they did to prep that slide is they did a longitudinal cut. So you're seeing the length of the fibers on that second image. So depending on how they prep the slide, it's going to look different under the microscope. Very, very important. Fibers we're going to see are often clustered right around the very precious vascular tissue that we're going to learn about. Vascular tissue is like the blood vessel system for a plant. Right? It's analogous to our blood vessel system. Very precious. And so that those fibers will tend to surround the vascular tissue to further protect it from getting damaged. Okay, so we'll talk about that. So we are going to get into the vascular tissue next, but we're getting close on running out. But here I like how it's kind of interesting how the artist drew the vascular tissue with blue and red, kind of like the human blood vessel system. It's kind of interesting that they took that color scheme. But again, it does drive home the point. It is analogous to our blood vessel. It's a way for the plant body to get nutrients and resources through the whole plant body. So when a plant is doing photosynthesis in the leaf, those sugars have to get all the way to the roots, right? And so the vascular system is going to help with transport. So we'll get into that on Thursday. All right.